Welcome to the C2Pipleline Innovation and Curiosity Center Sheep Eye Dissection Lab. By the end of this lab, you should be able to understand what the eye does, be able to identify the different parts of the eye and know their functions, and safely dissect the eye of a sheep. The human eye works much like a digital camera. Light is focused mostly by the cornea, which is the clear front surface of the eye that acts like a camera lens. The iris controls the amount of light reaching the back of the eye by automatically adjusting the size of the pupil, much like the camera's aperture. The eye's lens is located directly behind the pupil and focuses light. The lens helps the eye automatically focus on near and approaching objects, like the autofocus of a camera's lens. Light focused by the cornea and lens then reaches the retina, which is the light-sensitive inner lining of the back of the eye. The retina acts like an electronic image sensor of a digital camera. It changes optical images into electronic signals. The optic nerve then sends these signals to the visual cortex, which is the part of the brain that controls our sense of sight. Let's take some time to learn about the different structures of the eyes and their functions. Your diagram will help you to identify these structures as we move along. We will begin with the sclera and move in a clockwise direction to define the parts of the eye. The sclera is the white outer layer of the eyeball. At the front of the eye, it is continuous with the cornea. It is a very tough layer, as you will notice when we dissect the eye. This layer helps to protect the eye from trauma. The choroid provides oxygen and nourishment to the outer layers of the retina. Along with the ciliary body and iris, the choroid forms the uveal tract. The retina is a thin layer of tissue that lines the back of the eye on the inside. It is located near the optic nerve. The purpose of the retina is to receive light that the lens has focused, convert the light into neural signals, and send these signals onto the brain for visual recognition. Find the fovea on your diagram. The fovea is a dent in the retina that provides acute eyesight. The optic nerve is located in the back of the eye. It is also called the second cranial nerve. It is the second of several pairs of cranial nerves. The job of the optic nerve is to transfer visual information from the retina to the vision centers of the brain via electrical impulses. The central artery and vein bring necessary blood to and from the retina. The optic disc, also called the blind spot, represents the beginning of the optic nerve. The optic disc is also the entry point for the major blood vessels that supply the retina. The vitreous body is the clear gel that fills the space between the lens and the retina of the eyeball of humans and other vertebrates. The lens, by changing shape, functions to change the focal distance of the eye so that it can focus on objects at various distances, thus allowing a sharp, real image of the object of interest to be formed on the retina. The aqueous humor is a clear fluid in the front of the eye, unlike vitreous humor, which is clear jelly-like substance in the back portion of the eye. The aqueous humor is between the iris and the cornea, and it provides nutrients to the cornea. Find the pupil on your diagram. The pupil is the round, dark center of the eye. It opens and closes or gets bigger and smaller depending on the amount of light available. It controls the amount of light that goes to the retina. The iris is a thin, circular structure in the eye that's responsible for controlling the diameter and size of the pupil and thus the amount of light reaching the retina. Eye color is defined by that of the iris. The cornea shares this protective task with the eyelids, the eye socket, tears, and the sclera, or the white part of the eye. The cornea acts as the eye's outermost lens. It functions like a window that controls and focuses the entry of light into the eye. Muscles connected to the lens by suspensory ligaments adjust the shape of the lens to make it more or less curved, so as to increase or decrease the refraction of light. The ciliary body helps the eye with the process of accommodation, or the ability of the eye to change focus from near to distant objects, and vice versa. It also produces aqueous humor and holds the lens in place. Now that we have defined the parts of the eye and their function, we will begin our dissection. But first, a little bit about safety. During this dissection, you should be very careful with the tools as they are sharp. Never walk around with a tool in your hand. You should also wear gloves and safety glasses, a lab coat if available, and have fun. At this time, you will receive your eye and the materials necessary for the dissection. As this video continues, please do not perform any actions until the facilitator pauses the movie to give further directions. It is very important that you watch and listen to the video while it is playing. We'll pause the training so that everyone can obtain their materials now. Examine the front of the eye and locate the eyelid, cornea, sclera, the white part of the eye, and fatty tissue. We will pause the video now to make sure that everyone is able to locate those parts of the eye. Now let's take a look at the back of the eye. You should look for the optic nerve. If the optic nerve is not visible, use a probe to move the fatty tissue around until the nerve is exposed. We will pause at this time to locate the optic nerve. When the video is paused, you will very carefully use your scissors to cut away the eyelid, muscle, and fatty tissue from both the front and rear surfaces of the eye. Be careful not to remove the optic nerve. You'll cut along the surface of the sclera until the tissues are removed and your eye looks much like the pictures. The sclera is very tough, so you do not need to worry about cutting into this layer of the eye. We will pause at this point to allow everyone time to remove the tissues. If you're wondering why your cornea has such a cloudy look, the cloudy look of the cornea is caused by the death of this tissue. It's transparent when the animal is alive. When the video is paused, put your eye in the dissection tray and turn it so that the cornea is on the left and the optic nerve is on your right. Pick a place to make a cut or incision of the sclera midway between the cornea and optic nerve, as shown in the picture. Use the point of your scalpel to make a small cut through the sclera. Fluid should ooze out of the eyeball when you have cut deeply enough. We'll pause the video to make sure that everyone has been able to cut through the very tough sclera. Now that you have made a small cut in the sclera, when the video is paused, you will insert the point of your scissors into the slit made by the scalpel and cut the sclera with a shallow snipping motion. Turn the eye as you continue the cutting action. Cut the sclera all the way around the eye. You will need to support the eye in the palm of your hand while you cut the sclera. Some fluid from the eye may ooze from the slit as you make this cut. We'll pause the video at this time to cut around the sclera. Arrange the two halves of the eye as you see in the left picture. When the video is paused, you will examine the semi-fluid vitreous humor that fills the central cavity of the eye. It is clear in the living eye, but might be cloudy in this specimen. The vitreous humor, along with the aqueous humor, helps to maintain the shape of the eye. The retina lines the back portion of the cavity of the eye and goes forward to the ciliary body. When the video is paused, use your probe to lift and pull the retina back from the underlying choroid layer, as shown in the photograph on the right side. The retina is only firmly attached to the choroid at one place. This is the optic disc or blind spot. Here the nerve fibers leave the retina and form the optic nerve, which is directly behind the blind spot. Let's pause to make sure that everyone has been able to follow along. When the video is paused, you'll use your forceps to peel the retina away from the underlying choroid coat. The retina should stay attached at the blind spot. The choroid coat is dark and relatively thin. You'll use your forceps or probe to carefully and gently separate the choroid from the outer sclera. Then you will be able to see the three distinct layers: the retina, choroid, and sclera. The choroid contains an extensive network of blood vessels that bring nourishment and oxygen to itself and to the other layers. The dark color, which is caused by pigments, absorbs light so that it is not reflected around inside of the eye. We'll pause to separate these layers now. The vitreous humor may have already come out of your eye. If not, when the video is paused, use your forceps and probe to remove the vitreous humor from the front hemisphere of the eye. It may take some time and effort as the semi-fluid material separates easily. It helps to turn the hemisphere on edge to use a scraping motion to remove the fluid. Try not to disturb the lens that is just below the vitreous humor. Let's pause while everyone removes the vitreous humor and takes a look at the lens. Now you should be able to see the lens, ciliary body, and suspensory ligaments. The normal lens is convex shaped and somewhat elastic. It is held in place by the suspensory ligaments that in turn join with the smooth muscle containing the ciliary body. When the smooth muscle fibers contract, the resulting force flattens the lens and the degree of bending of light rays is reduced. Relaxation of the smooth muscle results in a thickening of the lens and a greater bending of the rays of light. We will pause to make sure that everyone has been able to find the lens and ciliary body. When the video is paused, we'll remove the lens by pulling it free from its attachments. You'll observe the shape of the lens, its stiffness and opaqueness. Suspensory ligaments may also be visible along the edge of the lens. We'll pause to remove the lens at this time. After the lens is removed, an opening, the pupil, which allows light to enter the eye, is visible. The opening is located in the center of the iris. Observe the oblong shape of the sheep pupil. In humans, the pupil is circular. The back side of the iris can be seen just above the pointer in the picture. Part of the iris is being lifted by the pointer, but the iris continues all the way around the pupil opening. We will pause to look at these structures now. This concludes the sheep eye dissection lab. Let's pause to answer the questions on the screen.